Human biological form is a product of its molecular constraints. We rely on microscopic building blocks to act as structural anchors, allowing our muscles and tissues to snap together and maintain their integrity under physical stress. But these structures operate on narrow margins. A single genetic typo can transform a life-sustaining molecule into a lethal biological threat. In the neonatal intensive care unit, a newborn presents with profound lethargy. Their limbs move in rhythmic, bicycling spasms. The clinical team notes a distinct, sweet, maple syrup-like odor radiating from the infant's skin and urine. Across the hospital, in the clinical pathology laboratory, identifying a solitary data peak on a mass spectrometry readout within a 48-hour window is the first step in halting the metabolic cascade. At this scale, the precision of the laboratory dictates the clinical outcome. To understand why a measurement at the molecular level carries such weight, we have to examine the rigid physical geometry of the molecule itself. L-valine is an essential branched-chain amino acid. Because the human genome lacks the machinery required to synthesize its specific isopropyl side chain from scratch, we are entirely dependent on dietary protein sources, beef, seeds, and whey, to maintain our internal supply. Valine is the simplest of the branched chain triad, defined by a rigid V-shaped isopropyl side chain. Because it branches immediately at the beta-carbon position, the molecule is exceptionally compact and inflexible. This physical shape makes valine strictly hydrophobic. It violently repels cellular water. When incorporated into a new protein, valine dives away from the fluid-filled environment of the cell, seeking the dry, central core of the protein structure. This movement triggers a thermodynamic shift. As valine displaces water molecules, it increases the overall entropy of the surrounding fluid. The energy released by this process physically snaps the protein wireframe into its functional three-dimensional shape. This hydrophobic reaction provides the mechanical force required to lock human biological architecture into place, stabilizing the proteins that define our physical form. Following ingestion, valine is rapidly absorbed in the gut and delivered into the portal bloodstream. Most nutrients are filtered by the liver, but valine performs the hepatic escape. Lacking specific enzymes, the liver lets valine pass through completely unaltered. It floods the systemic circulation, heading directly to the skeletal muscles, which hold 75% of our body's supply. In the muscle, valine triggers the mTORC1 signaling pathway to build new protein. During intense exertion, the muscle can also oxidize the valine skeleton to produce succinyl-CoA, fueling the cellular mitochondria when glycogen stores are low. Evolution accepted the risk of bypassing the liver's filtration to ensure valine could act as an immediate, unfiltered metric of dietary intake, commanding muscle growth and energy balance in real time. While the muscles hold the bulk of our supply, the fraction floating freely in the blood is microscopic, maintained strictly between 120 and 300 micromoles per liter. This circulating pool eventually reaches the blood-brain barrier. To enter the brain, valine must use the LAT1 transporter. This is a shared gateway with a finite capacity, meaning valine must compete for a slot alongside other amino acids like tyrosine and tryptophan. This bottleneck is why high-dose sports supplements can be counterproductive. Flooding the system with pure valine isolates saturates the available gateways, effectively jamming the toll booth. By monopolizing the transporter, valine starves the brain of the precursors needed to synthesize serotonin and dopamine. As neurotransmitter levels drop, the user feels a loss of focus and lethargy, while unabsorbed valine in the gut pulls water into the intestines, causing acute distress. There is a sharp irony in this mechanism. Overloading a nutrient to achieve a physical boost chemically induces a neurological crash. While dietary choices can disrupt this balance, genetic failures from birth create catastrophic outcomes.
Pathologically, these failures occur in two distinct modes: structural and metabolic. Sickle cell anemia is a structural defect. A single mutation places a water-fearing valine on the hemoglobin protein's exterior. To hide from blood fluid, it latches onto neighbors, forcing millions of molecules to crystallize into rigid fibers that distort the cell into a sickle shape. Because the rogue valine is locked inside the hemoglobin chain, this is a diagnostic illusion. A standard blood test for free-floating valine will show perfectly normal results. as the molecule isn't floating in the plasma, it's built into the cell's architecture. Contrast this with Maple Syrup Urine Disease, or MSUD. This is a metabolic failure. Because the body lacks the BCAAs enzyme complex, it cannot break down valine at all. Toxic keto acids flood the blood, disrupting the brain's fluid balance. The brain tries to dilute these toxins by pulling water across the barrier, causing the tissue to swell within the rigid confines of the skull. For the pathologist, the distinction is critical. Sickle cell is a structural flaw. MSUD is a free-floating metabolic crisis that must be detected before the pressure on the brain becomes irreversible. In a suspected MSUD crisis, laboratories look for plasma valine exceeding 800 micromoles per liter, alongside a specific biochemical signature, the presence of isoleucine. Confirming this requires liquid chromatography tandem mass spectrometry, a tool capable of measuring the exact weight of molecular fragments. The machine ionizes the valine at a mass-to-charge ratio of 118.2. It then smashes the molecule with energy and weighs the fragmented piece. If that fragment drops to exactly 72.1, the laboratory has confirmed the identity of the toxin with absolute certainty. Treatment begins by exploiting the body's signaling pathways. The medical team administers high-dose intravenous glucose and insulin. Insulin is an anabolic storage hormone. When levels spike, it commands cells to open their transport doors. This forces the toxic, free-floating valine out of the bloodstream and into the cellular structures. This rapid shift clears the circulation and takes the osmotic pressure off the brain. This is a brilliant use of the body's natural storage mechanisms, employing them as an emergency brake against a lethal chemical flood. Even with advanced mass spectrometry, the result is only as good as the sample. If the blood is compromised before it reaches the lab, the precision of the machine is irrelevant. A postprandial surge, testing a patient who hasn't maintained a 12-hour fast, can inflate valine levels by 40%, triggering false alarms and unnecessary interventions. Hemolysis is equally dangerous. If a rough blood draw physically ruptures red blood cells, they spill their internal valine into the tube. This creates pseudo-hypervalinemia, where the adhens is tainted before the analysis begins. Laboratories defend against this with delta checks, automated systems that flag any shift of more than 80 micromoles in a single day as an impossible biological event, requiring immediate human review. Precision in medicine depends on uncompromising stewardship to ensure the integrity of the evidence. Diagnostic stewardship means using these tests appropriately. Valine panels are reserved for neonates with unexplained lethargy, genetic screening, or profiling advanced insulin resistance. In critical care, we must also recognize BCAA burnout. Severe trauma burns through the muscle vault's valine reserves, requiring targeted nutrition to prevent rapid wasting. Valine is both a structural anchor and a metabolic risk. It is the molecule that stabilizes our protein geometry and signals our muscles, but it is also a potential neurotoxin if our internal pathways fail. If our survival depends entirely on a tiny, water-fearing molecule we cannot even synthesize, we have to consider what other invisible dependencies are hidden in our biology. Our architecture is a delicate balancing act, constantly negotiated at the microscopic level.